In this online activity guide, students learn about the lab technique polymerase chain reaction, or PCR for short. PCR is used to make millions of copies of a small segment of DNA so that it can be analyzed or used for another purpose. If you are using one of BABEC's PCR labs, this resource will help deepen your students' understanding of what is happening during their lab. In Part 1, students watch a video of PCR at the molecular level without sound and make observations and ask questions about what they see. They also watch a video of a scientist setting up a PCR reaction in the lab. Next, students are presented with diagrams, information, and questions about the two major components of a PCR reaction, master mix and primers. In Part 3, students read about what happens during a temperature cycle of PCR and describe each of the three steps using non-scientific language. In Part 4, students observe the pattern of DNA copies that are produced from the original DNA segment after multiple cycles. Students then have an opportunity to put everything together in Part 5, where they click through a simulation of PCR and answer questions as they go. Finally, students consider the similarities and differences between the natural process of DNA replication that happens inside a cell and PCR, which is conducted in a lab using a piece of equipment called a thermocycler. Please visit babec.org to find a link to this activity guide and to learn more about available labs and lessons for your classroom.